Two patients walk into the same clinic on the same day with the same diagnosis, so they start the same treatment. And 6 months later, one of them is in remission, while the other one is back in the hospital. This is perhaps the most puzzling and the most frustrating things in healthcare. Not that we don't have good treatment, but we don't always know which good treatment belongs to which person. And the reason is something called heterogeneity. It's a fancy word for a simple truth. Your body is not the same as mine. And the way a drug molecule interacts with your cells, your organs, your immune system is as unique as your fingerprint. That's a problem I'm working on. I'm Yue, assistant professor of molecular pharmaceutics, and I aim to make those hidden differences visible. Because once we can see them, then we can stop guessing and start choosing. Let me tell you how I got here. My scientific journey is rather unconventional. I actually trained in fields that traditionally don't overlap with each other. However, looking back, every step added a new piece, and importantly, a clearer sense of purpose. I started in chemistry, and I was drawn to this fascinating idea that you can use chemistry to engineer therapeutics to achieve the right dose in the right place at the right time. And that brought me to North Carolina, where I trained with Dr. Zheng Gu, a leader in smart biomaterials, and learned to engineer these smart nanoparticles that can carry the right amount of therapeutics to the right site and release them at the right time. Then I watched one of those perfect designs leave the bench and enter a living body. That's when the clean lines started to blur. I realized that in order for this powerful nanomedicine to actually work to help people, we need to understand the complicated system they are entering, which is the human body. And that search brought me to Caltech, to Dr. Jim Heath, a pioneer in technology-driven precision medicine. In the Heath lab, I learned to watch the human body at multiple scales. And at the Institute for Systems Biology in Seattle, I learned to speak the language of big data and use it to connect dots into a clearer picture of the human body. And now you can see what's actually going on there. You can start choosing wisely. That's how we move from guessing to choosing. And when I visited the Department of Molecular Pharmaceutics here at the University of Utah, Department Chair Dr. Hamid Gandhari shared with me his vision for the future of pharmaceutics and the united departmental support behind that vision. I was convinced this should be the new home for my science. And that's the exact moment the digital pharmaceutics lab became real. Here in DG Pharma, we develop nanotechnologies, microfluidic devices, and computational tools to read our body organ by organ, cell by cell. And you might be thinking, okay, this all sounds cool, but how does any of this actually help an actual patient on a random Tuesday morning? Well, that's exactly the question I want to answer here tonight. Here is how things typically go. A patient starts out healthy, develops symptoms, receives a diagnosis, starts treatment, and wait to see how that goes, knowing that sometimes it works and sometimes it doesn't. Well, I envision a future where fewer of us are stuck in this, try this, wait, and see whether it works, and more people are getting the right treatment sooner. I organize our work around 3 verbs: profile, predict, prevent. Profile. After treatment, we look back to understand why it worked for some but not others. Predict. Before treatment, we look ahead to help find the right treatment for this particular patient, not just the average patient. Prevent. Before a disease even begins, we try to find early warning signs so we can keep people healthy instead of waiting for a crisis. Let me make that real. Profiling means learning from what already happened so the next person benefits. Here you are looking at a slice of glioblastoma, the most common and most lethal malignant brain tumor. Median survival, 12 to 15 months. One single dose of immune checkpoint inhibitors 2 weeks before the surgery gave some patients 6 more months. However, many did not respond. And it is our job to find out the differences. What's inside the responders' tumors that's missing in the rest, and vice versa. And then we turn these molecular clues into better treatment combinations and test them out in new clinical trials. So our next patient will have a better shot. Now, prediction. A patient is newly diagnosed. A clinician needs to come up with a treatment plan. Can we provide a clearer answer before the treatment even starts, fast enough that we can actually change what happens to this particular patient? Here is how that plays out in pediatric leukemia. In some cancers, genetics can often point us towards the right treatment. However, in some aggressive leukemias, there's no such clear genetic switch, so the decision is harder. One promising approach here is we can actually directly test the patient's cancer cells against different drugs and see which one works. The problem is that could take days to weeks. We are dealing with a highly aggressive pediatric condition. The disease is progressing very fast. Our patients are young children, so we cannot afford such a wait. That urgency is what drove our collaborations with teams at St. Jude Children's and Penn. Together, we developed something called micropharma, a small automated device powered by artificial intelligence. Micropharma reads protein signals inside individual leukemia cells and predicts which therapies are likely going to work best for this particular patient. All that is done within 4 hours, which means that our clinician will have an optimal treatment plan the same day the patient walks in. And here on the screen, you are watching one of our devices in action. Everything I just described happens on this credit card-sized chip I'm holding right here in my hand. Our team is working hard to bring this chip to every clinic so we can make precision medicine accessible to everyone. Now, if profiling is learning from the past and prediction is choosing better today, then prevention is a dream. Imagine a future where we can identify risks early before symptoms force us into damage control. Here is one approach we are particularly excited about. Your body does something quietly all the time. Cells secreted these teeny tiny particles called extracellular vesicles into the circulation. They're like mini FedEx packages sent out by your cells, and they carry molecular clues about where they came from and what's happening there. Carefully studying these tiny particles from healthy people over time, we are now building a very detailed picture of what healthy looks like. Then we watch closely how that picture shifts with real-world exposures. We're talking about diet, pollution, chronic stress, viral infection, etc. And the hope here is that we spot early warning signs and convert them into something actionable, so we can prevent that very first symptom from happening. And as you can probably tell, this work is high risk, high reward. NIH support, including the Director's New Innovator Award, helps us take this on. And more support like this means that our teams can move faster. We can test out our bold ideas quicker, and we can train the next generation of scientists who will help us carry this forward. Of course, none of this happened in just one single lab. The most meaningful work I've ever been part of has always happened at the boundaries, where engineers talk to biologists, where data scientists sit with clinicians, where different perspectives collide and something new comes out. Over the past 4 years here in Utah, I've built collaborations stretching across the nation and into Europe. That's how ideas turn into impact. I started with one question: why does the same treatment lead to such different outcomes? Standing here today, I still do not have the full answer. But I believe that if we measure the right signals at the right time, we can move medicine from best guess to precision. And every collaboration, every new tool, every student who joins this effort bring us one step closer. Thank you.